Yeah, so first of all, let me start by thanking the organizers, Joe and Michelle and Elizabeth and Tim and the whole Nemo and Broad kind of groups for this opportunity. It's a great group of collaborators with a really good mission. I'm really happy to be part of it. I'm going to speak for I guess I have till 12:45. That's right, Joe. And probably I'll use that much time. I guess it might give you in fact 5 minutes or so. This is going to be a pretty high level. Of course, we can't possibly describe all the tools and the entire ecosystem in great detail, and you'll learn more about that throughout the presentations. But I'll hope to outline kind of some of the major ideas and show you the major points. The BICCN was started. It started as a outgrowth of something called the Brain Initiative Cell Census Consortium, which was an early effort to study techniques and to prototype techniques for scalability to show that it might be feasible to do brain-wide profiling at a single cell level. This was work that happened prior to 2017. It was written up. There's a nice paper written up about it with Joe Ecker and Honghui Zhang, which describes the experiences that they learned from that. And I think it's well worth looking at that paper. When the BICCN was formed, it became clear that this was to be a big part of the Brain Initiative, and it's one of the largest funded aspects of the Brain Initiative, and it's something I think that Brain Initiative sees as as a source of accomplishment. The goals as stated for the BICCN, this is essentially coming from the RFA, which was that to generate comprehensive 3D common coordinate reference brain cell atlases that integrate data, molecular data, anatomic data, functional data, lineage data, ultimately in mouse, human, non-human primates. The BICCN primary focus is really on mouse, with kind of leveraging key kind of prototyping activities in the marmoset and the human. Although, as I'll discuss, there was a substantial amount of deep dive in the marmoset in the human to really cover the primary motor cortex as well as certain other cortical areas. And that formed part of the package that we'll talk about in part 2. This slide here just shows the kind of the landscape, the timeline. As I mentioned, the highlight classification through the BICCC starting in 2014, which seems like quite a long time ago now. And the BICCN, which started in 2017 with the goal. And the number of awards that have been made in this is grown, quite significantly to ultimately almost 60 different kind of fairly major awards as part of BICCN. And BICCN right now, we'll have a few brief other words to say about that later, is really just starting. The fundable awards are essentially either under discussion or in final review, and it's not quite begun yet. It will begin in the fall, but it's imminent. So the kind of ecosystem that we're talking about is sort of represented in just the highest level terms is sort of something like this, is that there are these various kind of collaboratories, the U Awards, which are essentially highly kind of, robust data generation pipeline based awards run as kind of consortia in their own rights in some ways, and then RF1s and RO1s, which do slightly more specialized but highly advanced kind of techniques. A big part of the BICCN was this kind of relationship between the brain cell data center and the archives, which we didn't know about each other initially when everyone was applying, but we came to learn about each other and have since enjoyed a very positive and kind of productive work experience trying to manage these data. There are the four primary archives, as you probably well know, are the NEMO archive, which we will be learning most about, and its relationship tools here. The Brain Image Library from University of Pittsburgh and Pittsburgh Supercomputing Center. The BOSS-DB, which is a sort of electron microscopy and ultra structural kind of database managed from Johns Hopkins, and the DANDY electrophysiology and sort of whole brain imaging database from MIT. Oops. I'm going to have to. Basically, we through the BCDC, we try to catalog this data. It's not done accomplished down at a true kind of tissue level, but it's still right at the moment. It's at a type of project level with increasing levels of resolution sort of being built in. Users can look up this data. Users can look up this data. They can basically search for data sets, get access to them through the various archives. And there's essentially pipelines that are that you'll learn about actually a little bit later, but that are done through Terra and the Broad, which are highly kind of functional pipelines for processing these data in a very systematic way. This is sort of a little bit kind of another view of kind of the awards by the network. You can see here that how these awards have increased over time, and different in their collaborative research agreements, etc., various cooperative things and supporting infrastructure. So the main thing is that here is that this strong kind of multimodal approach to the problem. And it's leveraging the strong kind of applications of single cell omics from RNA-seq, single cell, single nucleus RNA-seq, epigenomic ATAC-seq, methylome-seq, and various other kind of variants of that. There's also imaging kind of techniques from kind of tracing and antrograde, retrograde tracing, connectivity, and multimodal sort of applications such as patch-seq and related techniques, BAR-seq. All these were kind of brought to bear on the first set of kind of the first study in the primary motor cortex, what we'll talk about a bit later. But the important thing is that there are now working groups that are off doing a considerably broader profiling across various areas of the brain using these. And so it's really interesting, like, as often happens in projects, is that you start and things take a little while to get going. Then you propose a kind of prototype project, and the thought is the prototype project will be kind of knocked out very quickly so that you can do the whole thing. But what in reality what happens is that the prototype project takes a lot longer than you imagine, because there's all kinds of considerations of working together and cross analysis. And so the bulk of the time is spent doing the prototype. But as soon as that happens, the floodgates are opened. And now there's, essentially these next generation kind of profile activities have just taken off. And that's really what's happened, is that now, since this initial prototype, we've been able to kind of profile whole brain omics. We're doing major kind of profiling across MFISH. There's lots of different techniques happening in connectivity, et cetera. So it's all kind of exploding in some sense. The data that we manage, as I said, there is this ecosystem here, which consists of our brain data center together with these archives. The BOSS-DB archive, the electrophysiologic archive, has not really kind of fully kind of come online with respect to BICCN activities, because there's only really one award that's part of BICCN there, and they're just kind of it's a full-fledged functional archive, which is really well organized, but it wasn't really part of this initial kind of package of analyses. So again, we certainly want to highlight here the NEMO archive and the NEMO analytics, because those are things we'll learn somewhat more about here. This workshop is more focused on omics technologies. Through the work of Carol Thompson and her team at the Allen Institute, we collect all this information about investigators and their techniques, and I can make kind of these pictures here, because based on the work from their teams, that are essentially factorizations showing the major techniques, all the different kind of techniques involved in their variants, and who's doing what, as well as what species they're doing it in. Now I have another bucket here, because there's half a dozen or more other species that the kind of cross species group is looking at, but it didn't really make sense to show them all out. There's just a wide variety of further species. The main ones that have been involved in this are mouse, marmoset, human, macaque. Those are the ones that you can see that these techniques and these people are bringing to bear on that. This is when we first made this description of this ecosystem. I had a figure which was an earlier version of this, but it was surprisingly throughout the course of the BICCN and the BCDC, just how much sense it did make. Largely, this is an improved version of this architecture, but it really is reflective of our original kind of thinking of it. Is that the difference is that we didn't really know exactly who was doing what where when we made our request for the award, but that kind of we of course gained clarity on that over the time of the BICCN. But a primary aspect of this, of course, are the multimodal data generators, right? Producing this high resolution data, doing QC and QA on it, trying to map their data to the CCF, and doing analysis, which is relevant to describing their findings, primarily for the goal of publication, right? There are two really primary activities that in groups that go on here. One are these sort of collaborative analysis working groups, and these were active, strongly active in terms of developing their work for the original publication package, but also have become really kind of revved up in the new 2.0 analysis kind of things, where it's a very active kind of groups trying to, in particular for whole mouse brain and for analyzing a human data package. And also standards. We're trying to inject kind of QC and QA into the data. There is a concept of data levels, which I'll tell you about in a minute, and published protocols that come from the consortium. All this data is deposited in the archives. They go into this level 1 validated data sets that are QA, QC approved by the data generators. And there's this concept of data levels, which means more added structure and structural description to data sets as they are produced. This is something we devised early in the BICCN, the concept of data levels, but it really is a very useful paradigm for helping users to identify what nature of data set is most relevant to their interests and their problem. For example, someone who's studying single, kind of alternative splicing variants would go down to only the most basic kind of reads, right? They would probably want QC validated reads, but they might even be willing to do a certain amount of checks to even improve those methods. Someone who's interested in building an application showing how data can be different cell types can be viewed in different brain regions would probably want data that had been linked to the CCF and anatomically localized, right? Someone who is studying kind of the biology of the data would also want their data to be, you know, kind of quantified and to have features. They would want cell by gene matrices, etc. And so it goes like this, right? Increasing levels of organization. It's not linear in the sense that linked could come before featured, of course, but this is the way we've chosen it. And ultimately, integrated data sets that are purely representative of knowledge kind of learn. And it's helpful to label these data sets. So we try to label these data sets and provide them, you know, essentially feedback to the, oops, I think I'm missing I missed a slide here. It may be out of order, but I just want to comment about the levels. One more thing is that, so this is at a level of great generality here, but we actually are working with the individual data producers to produce and define much more specific and relevant definitions of their data levels, what it means by modality. And we've collected all that, and we will be, you know, providing that back to the community and to the NIH for reporting considerations. You know, so there's a lot to, you know, this data here. This is a bit of a kind of a statement here, then, you know, about just sort of the scale of NEMO. And maybe you'll see this in a later talk, but it seems general enough that I certainly wanted to pull it forward. This was it's essentially material that came from the publication package on October 6th of last year, is that as of September 2021, the NEMO archive contained, you know, over 850,000 BICCN files. And so this is really a resource for, you know, people to really dig into and to really have legacy value. Enormous data of enormous size, 241 terabytes, and there's been a lot of activity and a lot of usage of this data. You can see here, as I've just kind of specified what these levels are a little bit more. These are the general definitions of the levels. And I won't, for sake of time, I won't go into this. These slides will be, of course, available to anybody who's interested. But in particular, as I mentioned, how the different groups were defining different data levels. Here, you know, essentially, when they get an award, when a group gets an award using a technique, they fill out a kind of data levels form, which indicates what they, you know, how they define a data level. And we have been working with these groups to put more kind of precision to this and more structure. So a little bit about just sort of it's in this segment here about the you know, what the BCDC does and provides is that, you know, we have the front end. You can go there and learn about essentially everything I'm talking about today. That part of it is a teams oriented page, which gives a project description of all the awards. You can get the details about their grants here. You can get more details about their team and their investigators by touching there. There's a data access part, which enables you to go directly into the BICCN data sources in the archives. Touching upon this will take you directly into archive linkages. And here there's the basic kind of archives are sort of shown over here as well. There's a scientific component to it too, where if you kind of go to the scientific page, this is a kind of review of all the materials that were provided in the mini atlas package of October 6th. And each of these links here will take you directly to the archived supporting data where it can be found for the publication. And you can explore the papers and really kind of dig in. When you touch on, say, one of these links, it takes you into our data catalog, which summarizes you know, the different data sets that are available from the BICCN corpus, and who you know, generated them and what the characteristics of them are. And then from there, it will go into archive related kind of materials. there's all kinds of tools and analysis things, far too many in this short introduction to kind of go through, but I'll just tell you about a few of them. In addition to, there's pipelines, visualization tools, mapping tools, and various analysis tools. We will hear later in much greater detail about some of these terrific pipelines that the Broad develops in processing and for community use of this data. So I won't dwell any more on that. That's a topic to be coming up a little later. A major part of this is the CCF mapping, as I mentioned. Here, data is mapped, expected that we will be identifying kind of spatially localized samples to the appropriate brain region, layer, et cetera, of the thing. This is increasingly important as we get out of one specific region and start to do brain-wide structures. This is a work that's undertaken over many, many years, starting from initial work from Hongwei Dong through later work of Lydia Ning and other team members who really led this development of the common coordinate framework. I also want to highlight other tools that were available. That there's a really great set of resources called QuickKnee from the Bjali and lab over in Oslo and Leergarten. That they sort of developed kind of both 2D and sort of 3D registration techniques that enable mapping into the Allen CCF. And this is being used increasingly by a number of people, these tools. Lots of sort of several visualization kind of tools that are under sort of way here. We have the Brain Architecture Project that comes out of Cold Spring Harbor, azimuth mapping tools. There are others. Sorry about that, folks. I don't know. I can't seem to control this phone, but let me highlight 4 specific tools today that you will hear in considerably more detail here. These sort of very powerful tools, the EpiViz, the BrainOme, from Aaron McConnell's group, Catlas from the Salk, and MetaNeighbor from Jesse Gillis' lab, and that these are four things very germane to the kind of data that is coming into NEMO, and you will hear considerably more about that later. So let me spend my remaining time talking a little bit. I'm going to transpose right here now to another topic here. It's just sort of the overview of some of the findings in the BICCN publication package. And so in doing this, I wanted to just go back and reiterate this thought that, the bulk of the BICCN was spent doing this so-called mini atlas project. And we conceived of this as like it was going to be potentially a 4-month bang it out kind of thing. And this is not surprising. It's not atypical that for things take much longer, right? But it's important to realize that whereas it did take considerably longer to do this and to publish this for this one region, it really set the stage for this really dramatic expansion and explosion that made everything else just go considerably easier. So it's far more than the profiling of a single region in its accomplishments. One of the major points is that the single cell technologies, in particular molecular technologies, provide a very, very powerful kind of approach for collaboration. And all these groups are kind of interacting with each other, from the BICCN to HubMap, the Human Molecular Brain Atlas program, which is very well developed now, the Human Cell Atlas and all its international kind of things. And this is it's really set it a very kind of strong collaborative environment that the BICCN enjoys. So this was the cover here. The paper was it was a paper. The major paper, as I said, it was October 6th. It was published online, and this, what essentially it was a flagship paper together with about 11 kind of papers that went with, which were sort of both supportive and related results. They didn't all contribute to the flagship, but most of them were companion papers for the flagship paper. So then, the network as a whole, its goals in this, as I stated, are to to do this sort of large-scale census, similar to sort of a population census, that defined neuronal and non-neuronal cell types, to get their distributional properties, proportions, spatial characteristics and distributions, and to provide a classification that can act as a taxonomy for further reference, right? To compare with other species, to compare with other modalities, and to get this data, to the extent possible, mapped into a common coordinate framework. So that was really the kind of overarching goal, and this was really, achieved as part of this publication. The major results, if you had to state them without going into specifics just yet, about the paper, is that it really did reveal a kind of, standardized genetic kind of common molecular landscape across cortical cell types. And it really unified these modalities. That we learned that the transcriptome and the epigenetic approaches aligned far more better and provided far more kind of cross corroboration than we might have expected. Essentially, one could derive, even without cross comparison, largely derive the same taxonomies from these data. And there's an enormously powerful now kind of toolkit available for exploring the regulatory significance of the epigenetic data with respect to gene expression. We learned a lot about how the different modalities kind of corresponded, how electrophysiological data and morphological data helped to define the cell types and the variability of them, as well as demonstrating sort of novel types that were not necessarily thought to be part of the motor cortex. There was a major genetic toolkit developed as part of this, which serves to basically allow people to now conduct further experiments into kind of targeting these cell types. And this is work that is really still, that this is just even still taking off. There's considerably more work being done on this. And it also, enough cross-species work was done to really form a consensus taxonomy across the 3 species of mouse, human, and marmoset. And so this was like one of the figure here, just sort of demonstrating how these techniques were used across species. The primary techniques being the molecular techniques I've mentioned, as well as MERFISH, which we'll have a bit more to say about in a minute. The multimodal technique of patch-seq, where one can, with certain degree of throughput, do both morphological and electrophysiological profiling. One can do whole-scale morphology through techniques like mouse light and FMOS to really get this long-range structure, axonal structure of neurons, and to understand a key determinant of types, namely their connectivity and projection. There's the epigenetic landscape provided with connectivity by epi-retroseq, as well as a detailed look at the kind of tracing and how cell types in primary motor cortex in their projections were organized. And then there's this data is heavily analyzed, and it provides kind of this big corpus. Just by numbers, this is a kind of a thing that the Nature package, they developed a really nice, you probably have seen it maybe, but they developed a really nice kind of vignette describing the papers and their significance. And they talk about the 11 companion papers. You can go to if you go to the site, you can see this nice kind of vignette describing the entire package, describing the over 2.2 million cells, the massive number of 1,700 neurons been reconstructed, and others kind of, a lot of patch-seq data. And these data are all described, if you really want to learn more about them, in the companion papers. Just to say a little bit more about the approach, the important is that the synthesis through these companion papers, that each group was taking their approach, but through this mini atlas project, the goal was to really do cross analysis and understand how they really corresponded to each other. Really enabled, a characterization of the different types, enabled a really detailed understanding of this data and how to display it. So, you know, a little bit more detail about some of the figures in this paper. Really, initially, one of the first figures is the first figure, really, except for the kind of methodological figure, which is in the extended data, is really about showing how the transcriptomic and epigenomic data kind of aligns and leads to a common kind of characterization of types. And you can see on top the sort of UMAP sort of presenting kind of the alignment of this, the number of cells in this, and you know, we used methods and including kind of bootstrap kind of statistical significance methods to derive a common kind of taxonomy. This is a terrific paper that came from Iran, and Honghui and Shizhen Yao did this work. This was some of the first entry point to the paper, describing the common kind of intersection of these taxonomies. Genes that were different from across the different, you know, different kind of techniques and approaches, conserved markers, and different kinds, essentially really a description of how these different data sets related to each other. I'm going to skip here to the last figure of the paper, which really, before we go into some of the others, but this was really the last figure that showed how all the different different modalities could be integrated. And, you know, we get a taxonomy which is both validated across techniques and across species, a common kind of taxonomy across the motor cortex in the 3 species, as well as describing really essentially there's kind of key morphological cell type projections and related kind of connectivity. This picture here just shows how the variability of how the cell types align across the different kind of taxonomies, and it shows very good correspondence. So another major technique which was used here was the Merfish technique, and this is the spatial technique, comes from Xiaowei Zhuang's lab, and even we've learned more recently about how this is even being scaled up considerably more. But they did essentially a de novo defining of cell types in the motor cortex using Merfish, as well as studying their associative connectivity properties and their correspondence with the basic omics approaches. And there's a lot of, you know, a great deal at the subclass level, you know, extremely good correspondence, even below that to some extent, but very good correspondence with the molecular techniques. There's an entire companion paper that describes this work. Another paper from Andreas Tolius' lab here and Scala et al. Is this using the patch-seq technique for several hundred kind of neurons and doing all these reconstructions, studying their physiological properties and aligning them with the molecular kind of techniques. Through the patch-seq, you get a lower resolution kind of transcriptomic profile, a more limited set of genes, but it's enough to localize these and to really map and to study these and how they relate to the overall omics profile. So there's a companion paper about that, which describes their findings. And what some of the interesting findings there are really the more still more increased kind of diversity and the phenotypic variation, which is available in a laminar basis from these different neuron types. It's a complex problem for which various newer machine learning techniques have been brought to bear to really understand how these correspond. And we really need the techniques like patch-seq to really solve the problem. Genetic tools, as I said, genetic tools through the work at the Allen Institute, Basilitazic, and other people have developed to target these cell type lines, and these provide ways of studying further. Again, the primary accomplishment of this work is in a descriptive taxonomy of the cell types of the primary motor cortex. And it's functional to the extent that functional properties are observed through physiology, but the significance of these types remains to be elucidated. And it's really these genetic tools which will provide us a way for people to further target these studies and to go about exploring more. Another major part of this work was the anatomy work from Hongwei Dong and his collaborators, Giorgio Ascoli, Pavel Ostin, Julie Harris, and others. This was a really big project to sort of develop a comprehensive cellular resolution wiring diagram using kind of a viral labeled Cree driver lines and then tracer methods and imaging to essentially reconstruct these and study the projection, essentially projections. They can study, you know, in kind of slice, they can study the local structure and projections, and then to use other kind of tracer methods and kind of other genetic techniques to try to get a hold of longer range projections. And to essentially, the figure demonstrates and shows across the major subclasses of the IT, ET, CT, essentially the connection, the connectivity of these neurons and the percentages to which these were described across these different areas of the cortex at a pretty deep kind of anatomic level. And so this wired diagram is fully constructed in their companion paper, and this picture, this summary of it is presented in the flagship result. It really describes, again, more of the complex range of relationships between, you know, the transcriptomic and their projection kind of and connectivity targets. Another, like I mentioned, that the epigenetic work was really a major part of this work, and the DNA methylation studies, which are able to kind of really get a handle on what kind of are the potential targets for expression of these genes, to understand how they relate to expression patterns, and to understand sort of enrichment of various motifs that exist in the data set and defining the data set. You know, there were specific findings really elucidated the role of kind of different kind of projection types in the mouse kind of primary motor cortex. This sort of work demonstrated that mouse MOT actually have MY projecting and non-MY projecting types. And this was sort of novel information relating to other kind of cell types, such as the layer 5 ET types that was unknown or unelucidated in previous studies. There's not really time to go into that, but this was kind of really a deeper dive into this aspect of the data, which was heretofore unelucidated in primary motor cortex. I want to highlight one more kind of study here. There was really, this was kind of a figure that comes out of one of the companion papers. It's a topic which I really enjoy, and it's this work from Trygve Bakken and people from Ed Lean's team, describing the really kind of a deep species comparison of alignment of the transcriptome, single nucleus alignment across species. Now they've done this over, you know, a dozen different species essentially, and are in the works of putting together a really large scale kind of comparison. It'll make a terrific kind of evolutionary kind of study. But they find the genes that define these clusters. They find that many of the clusters are the same, although that several of the markers and the defining characteristics vary from species to species, indicating a different kind of regulatory kind of landscape to the data. And an exciting study. This provided a key part of the full validation of the cross-species validation, but they've taken it much deeper now. And anyway, there's the papers. Each of these, I've just shown the links here from the Nature site. you can go in there and, if one of these techniques interests you more than others as a data modality, you can dig right in and go to these papers and link and find and study them. Any of these 11 are really quite interesting. I've gone through some of them. I can't say that I've read them all in great detail myself, but the ones that were more I thought that I would be able to benefit from more. And this, you can explore these papers directly from the BICCN website here. And there's a section that enables you to go dig in. Carol Thompson will be talking later about, much more detail about Allen tools. And, there may be a little bit of overlap, but I won't dwell on any of that now. Let me just mention, I'm sure she'll talk about this, but one way but it's useful to mention it here, is that we built this cell type knowledge explorer application, which summarizes the cell types across the species. And this provides a very nice way of navigating the BICCN primary motor cortex data set. Big shout out here to Raymond Sanchez and Jeremy Miller, who did quite a bit of work on getting this ready. So Carol, I'm sure, will be telling you more about this later. And as a final ending up, because it's important to end on time, let me just say that we are now entering the world of the BICCN, the Brain Initiative Cell Answers Network, where we will try to use some of this knowledge and techniques that we have learned about in studying human and non-human primate. There is additional mouse work that will be happening, but the shift is much more strongly into human. And you are here, right about there right now. And so there's another essentially 5 years of terrific collaboratory work and stuff here. So I guess I have maybe 1 minute if there's any questions.